Good morning, everyone. I am welcome you all to this 13th day of CSIR Life Science Question Answer Discussion session. Before I enter into the session, please let me know, am I audible? Can you see my slides? Is it visible? Please let me know through the chat. Yeah. Okay, thank you so much. We will start the session now. Okay, yesterday we have discussed about the some part of questions, part C questions in unit 4, that is the cell communication and cell signaling. Today we are going to discuss the remaining some questions because as per their schedule we are having this yesterday, today, tomorrow we are going to discuss the unit 4 that carries the most important questions. Okay, that is the most important chapter in the CSIR syllabus. If you are studying the unit 4, you can cover the nearly 7 questions, so you can achieve the 28 marks. So this is our focus. Try to follow the questions. If you have any doubts, please ask the doubts in the chat box. Okay, the first question. Upon a ligand binding, cell surface receptors move laterally to be capped and internalized. Leishmania, a protozoan parasite, can use several receptors on macrophages to get internalized. One of them is a toll-like receptor, we know, that binds to lipophosphoglycan on Leishmania. Once internalized, the parasite destroy the phagosomes. Which of the yeah. I think we discussed this in yeah. Okay, fine, no problem. It's a similar question which we discussed in previous day. It's a similar kind of question. Which of the following treatment is Leishmania infected macrophage with result lowest parasite number in a macrophage? Okay. In this question, you want to understand the question first. As per their question, the statement is upon ligand binding. Okay, every cell communication is happen with the ligand and receptor binding. Ligands are the signal hormones, receptors are present in the cell surface or parasite surface. Okay, upon the ligand binding, cell surface receptors move laterally to capped and internalized. Leishmania is protozoan parasite can use several receptors on macrophages to get internalized. One of them is toll-like receptor, that is TLR2, that binds lipophosphoglycan on Leishmania. So here the binding matters are the toll-like receptor and lipophosphoglycan. These two are bind and make the Leishmania and macrophage attachment. Once internalized, the parasite destroy the phagolysosome. Which of the following treatment of Leishmania infected macrophage will result lowest parasite number in a macrophage? So treatment, which is the best treatment? Here they provided the 4 option. In that, C is the answer. The both beta MCD and ammonium chloride. Okay, whenever the Leishmania infected macrophage is treated with beta MCD and the ammonium chloride, that macrophages will resulted the low parasite number. So there is no production after the treatment. The cell that parasite productions become low. So this is the question. Yesterday we have question. We discussed the similar kind of question in a different manner. Another question: in tissues, cells were bound together by physical treatment between the cell to cell or between the cell to extracellular matrix. Following the are the some characters of cell junctions. Okay, which is the cell junctions? Here the cell junctions are, it's here the state, this is a statement. Here they provided the answer question here. Following is not correct. So this correct, no, it's not corrected one is the yesterday we made a same question. This is the adhesions junctions or cell anchoring junction connecting actin filament in a one cell with that in a next cell. Here the actin filaments are playing a role, and desmosomes are cell matrix anchoring junctions connecting with the actin filament in a one cell to extracellular. These two are not corrected. Then next question: the oncogene and the tumor suppressor gene are treated as a cancer critical gene. Okay, this is the statement. Oncogene and the tumor suppressor gene are treated as a cancer critical gene. This is the concept here. In this cancer critical gene, increasingly the powerful tools are now available for systematically searching the DNA or mRNA of cancer cells for either significant mutation or altered expression. We can to have the tool. This is also statement to identify independently an oncogene or a tumor suppressor gene. Which of the following would be the most convenient test? Here they are provided the statement: the oncogene and tumor suppressor genes are treated with a critical cancer critical gene. This is one statement. Another statement: the more powerful tools are available to understand the DNA and mRNA of cancer cells in a systematic view. In the systematic itself we can find out the mutation and alterations. Most convenience test. As per their statement, the first one, A, is the answer. Transgenic mice that overexpress the candidate oncogene and knockout mice that lack candidate tumor gene. Okay, here the having the two statement. One statement is that it's overexpressing the overexpressing the oncogene. That means whenever we are doing the studies with the mice, that mice expressing the higher number of oncogenes. And another option, that's knockout mice. You can we can take it out. We can't mutate the particular gene, that is the oncogene. If there is a no oncogene in the mice, that knockout mice that lack candidate tumor suppressor gene. Okay, there is a no gene, tumor suppressor gene. This is the most convenient test. Next question. Okay, for example, one more thing about this oncogene treatment. Whenever we are doing the cancer or this oncogenes and tumor suppressor genes, we want to isolate the cancer or tumor cell. For that, we want to compare the cancered cell and cancerless cell. Okay, for that only we are doing a research in with the mice. We want to inject the cancer cell in the first generation. The second generation, that mice carries the first statement, and second period of time that mice is knocked out with a particular gene. Okay, that gene is it's not there in a mice, that is knockout mice. in the comparing the expression of both knockout and the transgenic, we can calculate, okay, this transgenic animals carries the oncogene, this mice having the cancer effect, and now knockout means that is no effect. This will also help for us a control values. The large protein of a pathogenic bacterium has been enzymatically digested to generate a mixture of peptide. Next question is the large protein of pathogenic bacterium has been enzymatically digested to generate a mixture of peptides ranging in a size of 3 to 8 amino acids. Okay, here the peptides are carrying the nearly 3 to 8 amino acids in the length. Peptide mixtures were then administered in an experimental animal. So peptides are injected in the experimental animal to generate peptide specific antibodies. In order to develop diagnostics for the bacteria, the antisera were used for Western blotting to detect the bacterium antigen. Western blotting failed despite the use of wide range of antisera concentration. What is the most probable cause for the problem? Here their statement, they are taken on large peptide and injected into the animal. After the injection, I experimented after the injection there, so there is a foreign particle, right? So the animal system will produce the antibody against the peptide. So the antibodies are produced. After that are produced in the blood. After that, sometime they are collected the antisera from that animal. That antisera is added in the Western blotting to detect the antigen. If there is specific antigen is present, then we can get the antibody. That Western blot shown the failure result. Why it's shown up here? The answer for the question is a C. The peptide specific antibodies were not generated as they were not coupled to the protein carriers. Here two statements are there. Peptide specific antibodies are not produced, not generated. Here in this step they are added the 3 to 8 peptide amino acid length peptides injected. Our human system not producing the specific antibodies. Okay, human system, that the system is recognized there is something wrong, but we that didn't know that which foreign particle. So they are producing the common antibodies, not specific, not peptide specific antibodies were not generated as they were coupled to the protein carriers. Not in that protein carriers means for antigen prediction, that recognition, that want to recognize the peptide, that recognize are not able to coupled to the protein carriers. So, one more thing about this concept, this peptide injection. Whenever we are adding the peptide drugs or peptide detector molecule, that peptide should be in a larger mode. If there is in a smaller molecule, our body is not recognized as the diagnostic compound, or it's not recognized as the protein. Drug. Okay, whenever we want to we want to the drug want to be attached on the protein, means that should be having the nearly 10 to 15 amino acids. That peptides alone shown better effects. And one more thing about this question is, the mouse was primed with the trinitrophenyl lipopolysaccharide. The mouse was primed with the trinitropolysacharide, that is TNP-LPS, whereas another mouse was primed with TNP, keyhole lymphet hemocyanin. We all know this is our protein. Yesterday we discussed about this keyhole lymphet hemocyanin. After 3 weeks, these mice were sacrificed and splenic cells were fractionated to B cells and T cells. B cells from TNP-LPS, that is the trinitrophenyl lipopolysaccharide, is primed mice were co-cultured with the T cells from the TNP-LPS or TNP lymph hole, I mean keyhole lymphat hemocyanide. Here the second step is, first step is they are after they are injecting primed means that is injected with these two compounds TNP-LPS, TNP-KLH. After 3 weeks, the mice is sacrificed and they are collected the spleen cells. To fractionate, they are collected the B cells and T cells. The B cells from the TNP-LPS is then primed mice. That LPS primed mice were co-cultured with the T cells from TNPL or TNPL-KLH. Okay, here the two mice are there, or it may primed with any of one. Similarly, B cells from TNP-KLH primed mice co-cultured with the T cells from TNP-LPS and TNP-LHS. Here you want to understand we have four co-cultures. What are all the four co-cultures? Here they ask the co-culturing. Okay, which is the co-culturing? Next thing here, one thing. Okay, first step you want to understand the mice carries these two. Okay, then the mice were sacrificed. Here just if you have pen and paper, draw it by yourself. You having the two first step, the bees first step itself we are having the uh, injection with the TNP and LPS. After the TNP and LPS, that mices were one mice is the both mices were sacrificed, and that T cells and B cells were taken. Okay, here the both cells are T cells and B cells were separately we are having, and first statement in this step is the B cells from, okay, where is the, I'm not able to move the mouse. B cells from TNP LPS primed cells were, okay, first we want to take the LPS. Okay, LPS in the LPS having B cells, right? We want to take that B cells and co-cultured with T cells from LPS, okay, or the KLH. Next thing about the statement is similarly B cells from, okay, in the both B cells, okay, LPS and LH, we are taking the B cells from TNP LHS taken from and co-cultured with the T cells from both LPS and KLH. Now we having the B cells of the both things, and then we are going to culture with the T cell, or it may be with the T cells of, here also T cell, here also T cell, T cells of LPS or KLH. So first taken a B cell, so here the mention we are mentioning as a B cell. And second thing, that is co-cultured with the T cell, so it's mentioned as here as a T cell. Then in the upper button, here first thing we want to take it from a TNP KLH. Why they mention as a KLH means, in this first step also we are having a B cell of LPS, and this LPS is playing a role in the primary step. But another step where the interaction, after the interaction, the co-culturing, that are interacted with LH. So the second step, the combination, this combination is produced with the second step. First step is the primary one, then final step is the output. That output having the KLH primed mice. It's a very notable thing here. Okay, next thing is about primer. The P24 uh, is the important core protein of HIV. P24 is an important protein of HIV. This protein is abundant during the active replication of virus. This is a statement. The serum of HIV patients was examined for the presence of P24 and antibody against P24 for proper diagnosis of the infection stage. Okay, for the diagnosis, they are using the P24. Match the clinical observation in the column A and B. So here they are provided this clue. That clue is P24 is a most abundant core protein which helps for the replication. So this is the clue. And for diagnosis, they are collecting the patient sample, patient serum, and they are checking the presence of P24 and antibody against P24. For here, they are asking the question match the clinical observations in column A and column B. The column A, that is here you can see this is the C, right? The option is a C. Option as per the option, P24 is present in a serum, that is the earlier stage of infection. Okay, that is the earlier stage. Why it is earlier stage means once it infected the human, our human system is produced the uh, it may produce the antibody against that. But this is the core protein of the HIV. So HIV protein just enter into the human cell, that is earlier stage. So our human system is not produced the antibody against that. This is the initial stage because they mentioned the P24 is a core protein. So if the sample, the serum show alone means that is the earlier stage. Second is a anti-P24 antibody is high in serum. Okay, antibodies are highly produced. That is the viral latency. Latency means that is the higher infection stage. Okay, our human system is it's actively against play actively playing a role against the particular antibody. So, if the serum contains anti-P24 antibody is high in serum, that is viral latency. That stage is initial death stage is started. If the antibody P24, the anti-P24 antibody beginning to decline with the corresponding increase with the uh, P24. It's a very important statement. Anti-P24 antibody begins to decline with the corresponding increases in P24. So the serum the P24 count become high, anti P24 antibody become low. So in this stage, what is the meaning means the progression of HIV from latency to lytic stage. Okay, that HIV it's going to the lytic stage, that recognize the particular antibody and that become that lytic the that or proteins. So once again, I repeat, once the P24 alone present in the sample, that is the earliest stage of infection, there is no immune response is produced in the body. Second thing is anti-P24 antibody is high in a serum, that is latency, the viral latency period. Then the anti-P24 antibody it become declined, that is become reduced in serum with the corresponding increases in the P24. The P24 become high. This case, the progression of HIV from the latency to lytic stage is high. These are all the about this concept. Okay, next is very important concept in the this fourth unit. Many of the questions are carries questions like this. Okay, you can without this band image, you can't see any CSIR question. All the CSIR questions they are asking question like this. Okay, mouse IgG is left either intact. Left lane is a here D. Here you can see the ABCD. Digest with the pepine or pepsin are treated with the mercaptoethanol and run a non-inducing STS page with and stained with the Kumasi blue. In this case, you can see the concept. Okay, 1 minute. Excuse me, 1 minute, please. The thing about this kind of question, we want to understand the molecular weights. Okay, here the lane, left side of the lane is labeled with the markers. The lane in A, B, C, D, this is the lane, are digested with pepine, pepsin, are treated with mercaptoethanol and run on non-reducing STS page. These are all the things for ABCD. Okay, and stained with the Kumasi blue. In separate experiment, pepine digested products are immunoblotted with anti-idiotypic monoclonal antibody. This is the extra statement. The pepine digested products are immunoblotted with anti-idiotypic monoclonal antibody. For following the following four profiles are attributed to each of the state treatment. Okay, here they are asking the treatment. The pepine alone, it's the immunoblotted one. Here that is the keyword code. Here the answer, our question is answer for the question is A. First, I am going to explain the concept in the question. Okay, they are taken IgG. Okay, their protein is their protein of interest for the take. They take on the mouse IgG, that is the protein. That protein is digested with the peptine and mercaptoethanol and the non-reducing STS page. These are all the four digestive material, and these four digestive materials are respectively present in the ABCD four lanes. And then after the digestion that are stained with the Kumasi blue, in separate experiment tania, that is the separate experiment, that pepine is digested, that is the column called pepine is okay, pepine digested, A column, pepine is digested with the pepine digested products are immunoblotted with the anti-idiotypic monoclonal antibody. So don't know whether it is be in A or B or C. We want to get the perfect the answer. So after the answer, I will discuss it. A is the correct one. They, as per their data, the given data, they consider A is digested with the pepsin, B is digested with so pepine, C is digested with mercaptoethanol, D is digested with uh, the digested with the peptide and followed by the anti-idiotypic immunoblotting. Okay, in this concept, why they consider this D is a separate one. Okay, in the by this digestion, mercaptoethanol and peptide and pepsin, the immunoglobulin G IgG uh, become digested into three fragments. Okay, these three fragments are plotted in a different manner. And one more thing, the immunoblotting recognized only the larger protein molecule. The IgG structure itself, they are having the complicated immunostructure. That IgG, it's shown the after the fine digestion, we can't recognize the uh, that immunoblotting is not able to recognize the fragments. In this immunoblotting, it can recognize the FC region of the antibody. So we are having the two FC regions. That FC region is bound with the or the digested bound digested FC regions are bound with the immuno molecules. So only that D is shown two immunoblots, two bands. That is the D1. You can root the answer from the back. Okay, so last statement, the separate experiment is digested with the immunoplot. So in the IgG, that time that needs the immunomolecules for immuno binding, that needs the FC regions and antibody regions. Other cases, that molecules are fully digested in different portions. This is the output of the this question. The next question is a very easiest question. It looks big, biggest one, but it's very easiest one. We want to read and understand the concept. Experimental growth, epithermal growth factor is needed for a growth of almost all the cells. So the epithermal growth factor EGF is the most needed one. This EGF receptor is transmembrane protein having an extracellular ligand binding domain. Okay, this is having the extracellular ligand binding domain, a transmembrane domain and cytosolic domain. There is a three domains are present in the EGFR, that is the extracellular epithermal growth factor. So this is the statement. After the statement, uh, that cytosolic domain is the protein tyrosine kinase domain. Okay, the cytosolic domain of the EGF is protein tyrosine kinase. Binding of EGF to receptor activity actives the protein tyrosine kinase, resulting in activation of transcription factors through intracellular transducers. So the binding of EGF to the receptor actives receptor. Okay, if it recognizes any receptors that activating the PK, the protein tyrosine kinase, that activates the transcription factors through intracellular transducers. In a cell type A, okay, hereafter the question will start. The cell type A, much of the intracellular ligand binds domain is deleted by the protease. In the cell type A, much of the extracellular protein binding domain is deleted by protease, such that the cytosolic domain of the protein tyrosine kinase become constitutively active, whereas cell type B is having a normal EGF receptor. This is a statement. Once again, I will repeat. Cell type A, much of the extracellular ligand binding domain is deleted. So there is no extracellular domain in the EGF. That is deleted by the protease, such as cytosolic domain of cytosolic domain of PTK become constitutively active, whereas the cell type B is having a normal EGF receptor. So now in a cell type B that having the normal EGF receptor, there is no deletion. What is the question? What will be the best fit graph for the growth of cultures of cell type A, B is complete medium in a presence and absence of EGF. So if the cells, the one here they are mentioned, the both cells are, if they may absent or they may present, two cases are there. Here the question, if there is a uh, the cells don't having the EGF means that cell number is become low indices, they are not able to transcribe. This is the statement, very very important, that not able to transcription activating the transcription factor. If there is a loss of EGF, that not able to produce the transcription factor, so that B cells are not able to producing the signal. Once again, correlate this statement. They are playing a vital role in the transcription. And also this read this factor. Okay, domain, the cytosolic domain of the active virus having the normal EGF, otherwise it not work out. But A is no, A is working in a normal working, but this is not able to produce. So as per their correlation of the statements, the B cells are don't having EGF, that not after the particular time, that is not shown any division in the cell, so that not there is a no cell improvement, cell number improvement, that a fully declined line. Number of the, so the as per the answer, then if there is a positive, if the time increment, the number also increase. And type A, that is it's positive is it's an upper layer. Why? Because the positive cells are highly increasing the transcription, but the negative factor, that there is no EGF, means there is a small number of the increments in the cell number. Okay, next question is this is also very important and easiest to concept. In a CSIR question, you can face the kind of question in a easy manner. Just read and imagine and understand. This is the whole concept in this kind of question. They must ask this question, this kind of the expression of protein related question. And this question is very, they must ask this kind of question. A particular type of cancer cells undergoes apoptosis by both extrinsic and intrinsic pathway. Now apoptosis, we have two pathway, that is one is intrinsic and apoptosis. The particular type of cells undergoes the both intrinsic and extrinsic pathway when treated with the chemotherapeutic agent X. The drug is X. The caspase 8 and caspase 9 are interior caspase associated with the initiators caspase associated with the extrinsic pathway and intrinsic pathway respectively. So caspase 8 is the initiator of the extrinsic pathway, caspase 9 is initiator of the intrinsic pathway. As per the statement, now if caspase A is silenced, so caspase is silenced, intrinsic pathway is silenced in a cancer cell type by shRNA transfection. What will be the best fit graph for apoptosis scenario in the cancer cells when treated with the agent X? This is the images. White is cell before transfection, cell before the cell after the transfection. Here the transfection is happen with the 9 caspase 9. For this we want to understand the basic theory. Okay, basic theory. That theory, if the concentration of the caspase 9, okay, is in active mode, that drug become in that cells are, if the intrinsic path, that is the intrinsic pathway activator, caspase 9 is a intrinsic pathway activator. If that is in active mode, the apoptosis percentage become high. That is the theory. Okay, caspase 9 is in active mode, that intrinsic pathway become activated and it stimulate the apoptosis in the cell. So here they are transfused with the caspase 9 with the shRNA. So there is lack of apoptosis. This apoptosis, there is a lack of apoptosis after the treatment of the caspase 9 and caspase 8 in active mode. So there is a higher apoptosis happen. Very easiest method, easiest question. Just understand and we want to answer it. Okay, next question. After the success surgery on chemotherapy, the tumor of breast cancer patients subsides. However, after almost 5 years, the tumor relapses in a more aggressive manner, not response to the conventional chemotherapy delivered earlier. The following postulation were made. That is the made. Okay, the statement we want to understand: after the successive surgery and chemotherapy, the tumor of the breast cancer patients subsides. However, after the almost 5 years, the tumor relapse in the more aggressive manner. Okay, after do not respond to the, there is a relapse of the recurrence of the tumor is high manner, aggressive manner, does not respond to the conventional chemotherapy. They are not able to control it. So the following postulations were made. The made post are PQR. Chemo-resistant cells were persisting within the tumor even after the therapy. Yeah, right. Okay, chemo-resistant cells are persistent in the tumor. A population of the quiescent cells which under favorable condition formed to new tumor cells. Okay, that's stimulating the new tumor cells formation. High ABC, that is the ATP binding cassette transporter expressing cells persisted in the breast during the chemotherapy. Okay, these are all the reasons behind the cancer cell occurrence. Recurrence of the cancers is the reason. These are the two reasons for the cancer recurrence. Next question: the followings are experimental observation made on the treatment of B cells. This treatment of B cells, the main thing is they provided the statements and the four statements. Okay, from the above observation, which of the following is correct interpretation for the role of CD4 in a B cell? Okay, the noted word role, which is the correct answer for the role of CD40 cells in a B cell. So correct answer is the rescue B cells from death. Okay, they want to rescue B cells from death, and IG class switch to IgG1. Okay, the two things are happen. One is rescues B cells from death, and IG class switched to the IgG1. Okay, these are all about the unit 4th question, and tomorrow also we having some question. Welcome to tomorrow class, day 14 class. Don't skip any classes. If you have any questions, you can ask in chat box or you can ask in a Telegram group. Thank you everyone. We will discuss more in tomorrow class.